DNA carries the instructions for how to build you from a single fertilized egg. It also commands all of the processes of the cell and cellular reproduction. DNA is packaged into discrete structures called chromosomes. These chromosomes stay in the nucleus of our cells. When we see a chromosome in a textbook, it is usually shown in the shape of an X. Let's make sure we understand what it is we are seeing. A chromosome that is in the form of an X has gone through the S phase or synthesis phase of interphase, in which the DNA content of the chromosome has been doubled. The S phase occurs before mitosis or meiosis begins. This is a single chromosome that has two identical DNA molecules called sister chromatids. These sister chromatids are connected at the centromere. A chromosome in interphase prior to the S phase, or the DNA synthesis phase, the chromosome would consist of a single double-stranded DNA molecule. It would not look like an X. Furthermore, the DNA is also unwound and decondensed during interphase, so even if you could visualize it, it would not appear nicely packaged into tightly wound chromosomes. Chromosomes condense to their visible form during cellular division, and they become easily observable at this time under a microscope. Genetic variation is important for the survival of the species. In the case of a cataclysmic event, if there is a lot of genetic diversity among the individuals of a species, there is a higher probability that at least some of those individuals will have the right genetic combination to survive the event. Here are some examples of genetic variation and its benefits to humans. Some humans have been found to have a mutation in their CCR5 gene, which gives them an immunity to certain strains of the HIV virus. Some humans carry a mutation in the actin-3 gene that gives them extra sprinting speed through a superior control of their fast-twitch muscle fibers. And some humans have a mutation in their AS3MT gene that allows them to tolerate levels of arsenic that are 80 times higher than would kill a normal person not having the mutation. Genetic variation is due to meiosis, which is a form of sexual reproduction. Genetic variation is caused by a couple of key processes that occur in the cellular reproduction of gametes. These are the eggs and sperm, and this cellular reproduction process is called meiosis. But in order to understand how we get genetic variation in meiosis, it is important to first remember what we learned about mitosis. So let's do a quick review. Somatic cells of our bodies reproduce using mitosis. The somatic cells reproduce to allow you to grow. Remember, you started out as a simple fertilized egg, and now you are made up of trillions of cells. All of those trillions of cells were created through mitosis. Mitosis is needed for growth and to replace old, damaged, or worn-out cells. Mitosis is a form of asexual reproduction. In mitosis, one single cell, called the parent cell, will split into two new cells that we call daughter cells. In mitosis, the daughter cells are genetic clones. In other words, they're genetically identical to the parent cell. There is no exchange of genetic material and no recombination of genetic material in mitosis. This is an example of a human karyotype. Somatic cells contain two sets of chromosomes. One set of chromosomes is inherited from the mother. The other set of chromosomes is inherited from the father. Somatic cells are called diploid. Di means two, and ploid comes from ploidy, which means the number of sets of chromosomes. Somatic cells are diploid because they have two sets of chromosomes. The human genome contains 23 pairs of chromosomes. In karyotyping, we can visualize the chromosomes by extracting the genetic material from a cell during metaphase when the chromosomes are condensed and easier to see. Then we can stain them and lay the individual chromosomes from mom and dad out side by side in what we call a karyotype. So we have two sets of 23 chromosomes, which means we have 46 chromosomes total in each of our somatic cells. So a cell that is diploid has two sets of 23 chromosomes for 46 total chromosomes. And a cell that is haploid has one set of 23 chromosomes, which would only be 23 chromosomes. Hopefully you got the math there. Did you notice anything strange about chromosome number 23 in the karyotype? The 23rd chromosome is not numbered. Instead, it gives two choices with an X or a Y indicated underneath it. This is because the 23rd chromosome is known as the sex-determining chromosome. If you are female, you will carry two X chromosomes, one inherited from your mother and one inherited from your father. If you are male, you will carry one X chromosome that you inherited from your mother and one Y chromosome that you inherited from your father. Since females carry two X chromosomes, and the egg comes from your mother, who is female, the egg must have carried an X chromosome. Since males are XY, the sperm could be carrying either an X or a Y from the father's genome. So the gender of the offspring is determined by whether the sperm carries an X or a Y chromosome. All of the cells of your body began as a single cell, a fertilized egg called a zygote. When a zygote forms, it forms from one complete set of 23 chromosomes from mom that it gets from the egg, and it also forms from another full set of 23 chromosomes from dad that it gets from the sperm. During fertilization, the sperm donates its set of 23 chromosomes to the egg. The zygote grows through the type of cellular division called mitosis. Each cell division brought about by mitosis yields daughter cells with the exact same genetic makeup as the parent cell. This is why each of your trillions of cells in your body, not including the gametes, have the same set of DNA. Gametes must be haploid for zygotes to be diploid. All of the cells of your body began as a single fertilized egg called a zygote. In order for a zygote to have two complete sets of chromosomes, no more and no less, the eggs and sperm that come together during fertilization have to be haploid. All gametes, eggs or sperm, are haploid. The word hap comes from the Greek word haplous, which means single, and ploid comes from the word ploidy, which means the number of sets of chromosomes. Gametes versus somatic cells. These other cells of your body, the non-gamete cells, are called somatic cells. The word soma is Latin for body, so these are the cells of your body. The eggs and sperm are made in a different way than the other cells of your body. Eggs and sperm are gametes and they form through the process of meiosis. The rest of the cells of your body, the somatic cells, undergo cellular reproduction using mitosis. In mitosis, one parent cell will produce two daughter cells. In meiosis, one parent cell will yield 4 daughter cells. Mitosis is used for non-gamete cells or somatic cells. Meiosis is used for gametes, which are the eggs and sperm. In mitosis, the daughter cells or offspring are genetic clones of the parent. In meiosis, the offspring or daughter cells are genetically unique from the parent cell. In mitosis, the daughter cells that are formed are diploid, meaning they have 2 sets of chromosomes, one from each parent. In meiosis, the daughter cells that are formed are haploid, meaning they only have 1 complete set of chromosomes. Mitosis is a form of asexual reproduction. Meiosis is a form of sexual reproduction. How does genetic variability occur? The short answer is through the processes of crossing over and independent assortment. But now I've probably left you with even more questions, so let's clear this up. Here is a schematic of the phases of meiosis. We have meiosis 1 and meiosis 2. So you might notice that this looks a bit familiar because the phases of meiosis are named after the phases of mitosis. There are some differences though. Meiosis, you can think of it as kind of like 2 consecutive rounds of mitosis, but there are some significant differences. In late prophase 1 of meiosis, homologous chromosomes will pair up at the middle of the cell. This is done in preparation for a process called crossing over. For example, chromosome number 1 from mom is going to pair up with chromosome number 1 from dad, and so on. There are 2 events that take place in meiosis 1 that cause genetic variation. They are crossing over and independent assortment. Crossing over. These homologous chromosomes will then swap genetic material between the maternal chromosome and the paternal chromosomes. The entire process is called synapsis, and just the point at which genetic material is swapped is referred to as crossing over. Afterwards, each chromosome is then a mixture of both maternal and paternal DNA. The other way in which genetic variability is accomplished in meiosis is in independent assortment. Independent assortment takes place during anaphase 1 of meiosis. In independent assortment, or anaphase 1, we see that homologous chromosomes are now going to separate. They will be pulled towards opposite sides of the cell. And remember, crossing over has also already occurred. So each chromosome has a combination of DNA from both parents at this point. However, it's absolutely random which chromosome will go to which side of the cell. This is why it's called independent assortment. After anaphase 1, the cell splits into 2 non-identical, genetically unique daughter cells that are haploid. Each chromosome consists of 2 sister chromatids. In meiosis 2, the chromosomes line up during metaphase 2, and the sister chromatids will be pulled to opposite sides in anaphase 2 to create a total of 4 new gametes at the conclusion of telophase 2. The 4 gametes are haploid, and each of the chromosomes contains only 1 molecule of double-stranded DNA, so it does not look like an X. Let's explore the patterns of inheritance. A gene is a segment located on a chromosome that holds the genetic instructions for a specific trait. An allele is the type of gene or form of a gene that a specific person has. For example, two people have genes that code for eye color. One person has an allele for brown eyes, and the other person has an allele for blue eyes. Since you carry two complete sets of chromosomes, one from mom and one from dad, you carry two genes or alleles for each genetic trait. So, what determines what trait you will display? A genotype is the actual genetic makeup that a person has for a trait. The genotype will include both types or alleles of the gene that the person inherited from both the mother and the father, whether or not they show the trait. A phenotype is the outward display or the physical expression of a person's genotype. It is not always possible to infer a person's genotype based solely on their phenotype. Dominant versus recessive alleles. Some alleles or forms of a gene are more dominant than others. A more dominant allele can mask the effect of a recessive allele. Let's say a man with one dominant allele for brown hair and one allele for blonde hair marries a woman with blonde hair, which is recessive. When explaining a person's genotype, it is useful to use the following terms. Homozygous dominant is a person possessing two dominant alleles for the trait. Homozygous recessive is a person possessing two recessive alleles for the trait. Heterozygous is a person possessing one dominant allele and one recessive allele for the trait. The genotype for this man is heterozygous because he has one allele that is dominant and one allele that is recessive. The phenotype of the man, however, is dominant. He displays the dominant trait of having brown hair. The genotype of the woman is homozygous recessive because she has two recessive alleles for hair color. The phenotype of the woman is recessive, which is blonde hair. Here is an example of autosomal dominant inheritance. A good way to work these types of problems is to make a Punnett square. The letter that you use is not important, but the case of the letter is. A capital letter indicates that it is a dominant allele. A lowercase letter indicates a recessive allele. Place the genotype of one parent on one axis of the Punnett square, and then the genotype of the other parent on the other axis of the Punnett square. Then you can simply fill in the table. In this example, which is autosomal dominant inheritance, we see that in autosomal dominant inheritance, 50% of the offspring will have brown hair. 50% of the offspring will have blonde hair. The 50% with brown hair will be heterozygous for the trait of hair color and will display the dominant phenotype. The 50% with blonde hair will be homozygous recessive for the trait of hair color and will display the recessive phenotype. Let's say we have a man and woman who are both carriers for the cystic fibrosis gene. The cystic fibrosis gene is recessive, so neither of the parents display symptoms. How many of their children might be affected and how? This would be a case of autosomal recessive inheritance. When you fill out your Punnett square, you will find that 75% of the offspring will be carriers of the cystic fibrosis gene, but they will not have the disease or experience any of its symptoms. But 25% of the offspring will inherit the cystic fibrosis gene from each parent and will display the symptoms of the disease. Sometimes traits can be linked to the 23rd chromosome, which is the sex-determining chromosome. Let's look at what happens in an X-linked recessive inheritance pattern. Color blindness is a great example of an X-linked recessive disorder. Let's say that a normal non-colorblind man has offspring with a woman that is a carrier of an X chromosome that has a gene for color blindness. Since women have two X chromosomes, a woman can only have the phenotype, or the outward display or physical manifestation of color blindness, if they inherit two X chromosomes for color blindness. However, a woman can carry the allele for color blindness and pass that gene on to her offspring. So let's see what happens in this situation. When we fill out our Punnett square, we will find that the male offspring will have a 50% chance of being colorblind, and the female offspring will have a 50% chance of being a carrier for color blindness. But you might also notice, because this is a recessive trait and it's X-linked, it is impossible in this situation for a female to have inherited two X chromosomes carrying the color blindness gene. Therefore, there is a 0% chance that any of the female offspring will have color blindness. Blood types. Blood types are an example of genes that can be codominant. There are 3 alleles or forms of genes for blood type. These are type O, these are type A, type B, and type O. Alleles for blood types A and B are codominant. That means if a person inherits an allele for type A and an allele for type B, they will have a blood type of AB. The allele for type O is recessive to both type A and type B alleles. A person inheriting one allele for type A and another allele for type O will be type A. A person inheriting one allele for type B and another allele for type O will be type B. A person inheriting 2 alleles for type O will be type O. Can you figure out what the genotypes and phenotypes would be? For the parents, see if you can guess the phenotypes for the blank circles before they are removed. The dad has an allele for type A and an allele for type O. Type A is dominant to type O, so dad's blood type, or phenotype, is type A. The mom has an allele for type B and an allele for type O. Type B is dominant to type O, so mom's blood type, or phenotype, is type B. Let's fill out our Punnett square. Don't forget, you can also try pausing to see if you can fill out the rest of the Punnett square. Here's an example of how two couples having the same blood types can produce different offspring. So let's try one with AB. So let's go ahead and fill out our Punnett square and see what we get this time. And remember, they're codominant. So this time, we will find that 50% of the offspring will be AB, and 25% of the offspring will be type A. Another 25% will be type B. In this situation, we see that 50% are type A and 50% are type B. None of the offspring are type AB or type O. Thank you for watching.